Okay, so let's unpack this. Imagine you're a scientist using these, really powerful tools in genetics, genome-wide CRISPR-Cas9 knockout screens.
Right, the ones that let you figure out what a gene actually does.
Exactly, by snipping it out and seeing what happens. It's a very direct measure. Quite different from just looking at gene expression levels, seeing if it's on or off,
hmm
but actually working with these screens and the huge amount of data they generate, well, it gets tricky.
Oh, it really does. A basic challenge right off the bat is that the little guide RNAs, the
Yeah.
sgRNAs, the things that tell CRISPR where to cut,
Yeah.
they just don't all work perfectly.
Yeah.
Their efficiency, like how well they actually knock out the gene, could be all over the place.
Oh, okay. So you can't just trust one guide RNA result for a gene.
No way.
Yeah.
To get a reliable read on what the gene's really doing, you absolutely have to use multiple sgRNAs for each gene. And then, well, you've got to figure out how to combine those different results effectively.
Right, that makes sense. And then there's another layer, isn't there? Like people run these screens in different kinds of cells.
Exactly. Or under different conditions, maybe with a drug treatment or without. Each experiment gives you what's called an sgRNA profile for that specific setup.
So you end up with lots of these profiles.
They do, and integrating them, trying to compare them, combine the data, pull out insights that hold up across maybe different cell lines or treatments, that's a pretty significant analytical hurdle.
You're left with this really complex, sort of multidimensional data puzzle.
That's a good way to put it. And those variations, both within a screen, because of the sgRNAs, and across different screens, they make it tough to really pin down reliable gene functions.
Okay. So how do you cut through all that complexity? Our deep dive today is looking at a research paper that explored something maybe a bit different.
Yeah, they looked at applying a mathematical technique, one you see a lot in data science, but maybe less commonly applied this way in genomics. Tensor decomposition.
Tensor decomposition. Okay.
And this paper specifically digs into how TD, this method for analyzing data with many dimensions, can tackle these exact CRISPR screen problems, the variable sgRNAs, and integrating multiple profiles, but doing it simultaneously.
Right. So our mission here is to explore this paper, get a handle on how tensor decomposition actually works in this context, see how it stacks up against the methods people are already using.
It's a pretty sophisticated methods, actually.
Yeah, and find out if TD offers any unique insights into, the structure of all this CRISPR data.
Okay, let's dive in.
So we've got the challenge set up, tricky sgRNAs, needing to integrate data across different experiments, these profiles. You mentioned the field already has tools for this. What were some of the existing ones the paper talked about?
The paper does note that researchers have come up with various ways to handle these issues. They list a few examples, like MAGE. Yeah, it uses statistical models. Then there's Bagel, which is more a Bayesian thing. Series takes gene copy number into account. And STARS is often used for finding genes that help cells grow faster, positive selection. So different approaches.
Different angles on the problem.
Yeah.
But they focused on one comparison in particular, right? The sort of state-of-the-art benchmark.
Yes, exactly. That's JKAS, DTKS. It stands for Joint Analysis of CRISPR-Cas9 Knockout Screens.
Okay, JAGS.
And the paper points to JAG-S as a leading method, mainly because it is designed to integrate data across samples and conditions. It also tries to figure out how active each individual sgRNA is using, well, a pretty sophisticated Bayesian model. It's powerful, definitely, but also quite complex under the hood.
Okay, so you've got this complex data and these specialized, fairly advanced tools like JAGS trying to make sense of it. And then this paper throws tensor decomposition into the ring. How is TD different? What's its main selling point here?
Well, fundamentally, tensor decomposition, or TD, it's a linear algebra technique. You can kind of think of it as an extension of something like PCA or matrix factorization, but for data arrays that have more than just two dimensions.
So like a data cube or even 4D?
Precisely. A tensor could be 3D, 4D, whatever you need. And TD basically breaks down this big multi-dimensional block of data into simpler component vectors. The paper presents it as, mathematically speaking, less complex than some of those other specialized genomic methods.
And TD has been used in biology before, just maybe not for this exact problem.
That's correct. It's actually got a track record for integrating different kinds of biological data. Maybe combining gene expression data with protein data and genetic variations to find patterns that cut across all those levels.
But the new idea here is applying it specifically to these CRISPR screens and how it puts the data together.
Exactly. That's the key innovation they're exploring. Using TD to integrate not just the data from the multiple sgRNAs trying to target one gene.
Which you need to do anyway.
Right. But also, crucially, integrating the data from the multiple sgRNA profiles,
Mm-hmm.
those different cell lines or conditions, doing it all at the same time, right from the get-go.
Ah, okay. So most other methods, like you mentioned, they might analyze each experiment separately first and then try to combine the final results.
Yeah, that's often the approach. Process profile A, process profile B, then see how the gene scores compare.
But TD takes the whole lot, all the profiles, all the sgRNAs for all the genes, and looks at it as one big high-dimensional thing from the start.
hmm That simultaneous integration seems to be the core difference, and potentially a big advantage. It might let it spot patterns that exist across the profiles more inherently.
Okay, that makes sense. So how did they actually test this? Did they use real-world data?
They did. They applied their TD method to 5 different publicly available sgRNA profile datasets. And importantly, these were the exact same datasets that were used in the original paper describing JCK-Cas.
Oh, smart. So they could do a direct head-to-head comparison. Which data sets were they?
Let's see, they used USA v1.0, TKO v1, Whitehead, GKO v2, and Havana. So good range of common screen libraries and data.
And structuring this data for TD, how did that work? You said tensors, so multiple dimensions?
Yeah. Typically, they organize the sgRNA count data, basically how many times each sgRNA showed up after the experiment, into a 3D tensor. The dimensions would be genes, the specific sgRNAs targeting them, and the different profiles, like the cell lines or conditions.
Genes, sgRNAs, profiles.
Oh,
Got it.
and for a couple of the data sets, GCOE2 and Havana, they actually had biological replicates for the experiments. So for those, it became a 4D tensor, adding that replicate dimension in.
And they also played around with the type of numbers they put in the tensor, right? Not just the raw counts.
Yes, exactly. They tried TD using both the raw sgRNA counts and using logarithmically transformed counts. Log transformation is super common in biology data analysis, often helps normalize things, so they wanted to check if TD needed that step or if it could handle the raw data just fine.
Makes sense. And then the big test. Could it actually find the important genes? How do they measure success?
They used a pretty standard approach for this kind of study. They relied on what are called gold standards, basically. Well-established lists of genes known to be essential for cells to survive, and other lists of genes known to be non-essential.
Okay, the must-have genes versus the dispensable ones.
Right. And they looked at the parts of the TD output, the components specifically related to genes, and assessed how well those components could tell the difference between the essential and non-essential groups.
How did they measure that difference?
Their main metric was the area under the ROC curve, or AUC. It's a standard way to measure how well a model can classify things into 2 groups. An AUC closer to 1 is better. And then they directly compared the AUC scores they got from TD to the scores JACAS achieved on those same data sets.
All right, moment of truth time. How did this supposedly simpler linear algebra thing actually do against the sophisticated state-of-the-art JACAS?
Well, the main result is pretty striking, actually. Tensor decomposition did successfully distinguish the essential genes from the non-essential ones, and its performance level was consistently competitive with JACAS across all 5 of those data sets they tested.
Competitive, so like similar AUC values.
Yeah, pretty much. The paper shows tables where the AUCs for TD were often hovering around, say, 0.77 to 0.81, and JACAS on the same data was maybe 0.79 to 0.85, so very close. The paper uses the phrase, quite comparative.
Wow. Okay, that is interesting. A simpler method basically keeping pace with the top benchmark. But I remember reading something even more surprising about data sets without controls.
Yes, this is where TD really showed a unique strength. It managed to maintain that competitive performance even when analyzing data sets like GCOV2 and Havana, which, in the way they were processed for this comparison, didn't include explicit control samples.
What do you mean by control samples?
Like samples from the start of the experiment before gene knockout effects took hold, or samples treated with a non-targeting guide RNA. These are often used as a baseline.
Okay, so TD worked well even without that baseline comparison built in.
Exactly. And the paper notes that JCK-S, at least as typically used, couldn't actually be run on these data sets without having those controls available. TD somehow found a way to pull out the essential gene signal anyway.
That seems like a really big practical advantage if you don't happen to have perfect control data.
It could be, yeah. And they also looked at the ROC curves themselves, not just the AUC number.
The shape of the curve, what did that show?
They found that the shapes of the ROC curves generated by TD and JCK-S were remarkably similar.
Huh. What does that imply?
Well, it suggests that even though the math underneath is totally different, linear algebra for TD versus this complex Bayesian stuff for JCK-S, they might actually be latching on to similar underlying biological signals in the data that relate to gene essentiality.
So maybe hitting the same biological truth through different mathematical paths?
Possibly. It really is, as the paper points out, quite remarkable that something described as mathematically simple can compete so well with a highly tuned, specialized method like JAK-S, especially with that added bonus of working without controls where the other method couldn't.
Absolutely. Okay, so TD performs well, but did it also tell us anything new about the data itself, like how it was achieving these results or what patterns it was picking up on?
It did. They went beyond just the performance scores and looked into the components that TD generates. Specifically, they analyzed these things called singular value vectors attributed to cell lines.
Okay, singular value vectors. Break that down a bit.
Think of them as key patterns or directions of variation that TD finds within the data dimensions. So the cell line vectors capture patterns that are specific to each experimental profile or cell line used in the study.
Right. And they looked at which of these cell line patterns were important for finding the essential genes.
Exactly. They checked which cell line vectors were most strongly associated with the gene vectors that did the best job distinguishing essential from non-essential genes.
Okay. And for the data sets that did have control samples included, what was the link?
For those data sets, USA, TKOV1, Whitehead, they found something pretty intuitive. The high-performing gene vectors were linked to cell line vectors that showed a really clear separation between the control samples, or maybe the starting time point samples, and all the other experimental samples.
That makes sense. Good performance comes from being able to clearly see the effect of the knockout compared to the baseline control.
Precisely. It's capturing that core experimental contrast.
But what about GeCoV2 and EVA, the ones without controls? How did TD manage to perform well there? What were the important cell line patterns it found?
Yeah, this is the really fascinating discovery. For those data sets, the important cell line vectors, the ones linked to good performance, obviously weren't separating out controls because there weren't any explicitly included.
So what were they related to?
They found that these key cell line vectors were highly correlated with completely independent data about CRISPR efficacy attributed to cell lines. They actually downloaded this efficacy data from the DepMap database.
Whoa, wait. DepMap. What is that exactly?
DepMap is the Cancer Dependency Map Project. It's this huge public resource where they gather and analyze data from genetic screens, like CRISPR screens, across thousands of cancer cell lines. They map out which genes are essential for which cancers.
Okay, so it contains information about how well CRISPR generally works in different cell lines or how sensitive they are.
That's right. It includes data reflecting the inherent sensitivity or responsiveness of different cell lines to CRISPR manipulation. Some cell lines just show clearer knockout effects than others.
And TD, just by analyzing the screen data itself without any controls, managed to identify cell line patterns that match this independent measure of CRISPR sensitivity.
Exactly. It suggests that TD was able to pick up on this fundamental biological property of the cell lines, how amenable they are to CRISPR screening directly from the experimental data. And importantly, this inherent property seems linked to how well TD could then identify essential genes in those lines.
That's pretty cool. It's like it found a relevant biological signal on its own. What about the other big variable, the sgRNAs themselves, the ones with different efficiencies? Did TD offer insights there?
Yes, this was another really key finding related to how TD works. They looked at the singular value vectors attributed to sgRNAs.
Okay, same idea. Patterns related to the sgRNAs.
And they consistently found that the first sgRNA vector, the one capturing the most dominant pattern across all the sgRNAs, had by far the largest contribution to the overall structure that TD found. Its corresponding value in the core tensor was largest.
The first vector was most important. What does that tell us about how TD handles sgRNA variability?
It strongly suggests that TD is actually quite robust to those varying efficiencies of individual sgRNAs. It seems like instead of needing to meticulously model how well each specific sgRNA works, which is what complex methods like jCNAs try to do,
Right.
TD appears to succeed by leveraging a sort of dominant shared signal, or maybe an average effect, across the multiple sgRNAs targeting the same gene. It kind of captures the main trend without getting bogged down in the noise of each individual guide's quirks.
Huh. So maybe its mathematical simplicity in dealing with the sgRNAs isn't a weakness, but part of why it performs so well, it just focuses on the main effect.
That's definitely a possibility the paper raises. This ability to effectively use the sort of uniform effect captured by that first sgRNA vector might explain how it competes with methods that use much more complex sgRNA modeling.
Fascinating. Okay, let's try to pull this together. Based on what the study found, what are the main advantages of using TD for this kind of analysis?
Okay, summarizing the pluses. A big one is that it seems to be the first method designed to integrate multiple sgRNA profiles right from the start, simultaneously.
That simultaneous integration.
Right. It achieves performance that's competitive with state-of-the-art methods like JESIC-SS, but using what's described as a much simpler linear algebra approach.
Simpler math, competitive results.
Mm-hmm. And crucially, it works well even when you don't have explicit control samples, apparently by identifying other relevant biological signals like that cell line efficacy. It also seems pretty robust to the variable efficiency of individual sgRNAs, potentially simplifying things.
And there was that point about data transformation, too.
Oh yeah, for Oven, the biggest data set, using the raw count data worked just as well as using the common log transform data, which definitely makes you question if that standard pre-processing step is always needed, at least for TD.
Okay, those are some strong advantages. Did they identify any downsides or limitations to the TD approach?
The main one mentioned is a practical constraint. Because TD needs the data structured as a complete tensor, like that 3D or 4D block, it requires you to remove any genes or cell lines that don't have enough associated data points, like enough sgRNAs measured or enough replicates.
Ah, so you might have to filter out some data beforehand if it's patchy.
Yeah, potentially. You need a reasonably complete data matrix or tensor to run the decomposition effectively, as applied here.
Okay, got it. So we've really dug into this paper, the problem with CRISPR data, the existing tools, how TD works, its surprisingly good performance, and the neat insights it gave. Let's connect this back out. Why should you, the listener, care about this, whether you're in genomics or just interested in data analysis?
Well, for anyone working with or benefiting from functional genomics, better ways to analyze CRISPR screens are huge. It means potentially faster, more reliable discoveries about which genes are crucial for, say, cancer cell survival, or which are involved in a particular disease process.
Which leads directly to identifying new drug targets, understanding diseases better, accelerating the whole research pipeline.
Exactly. And I think this paper is also a really nice illustration of something broader, how a technique that might seem kind of abstract tensor decomposition from mathematics can be applied in a really clever, novel way to cut through incredibly complex biological data.
It suggests that sometimes maybe a simpler mathematical tool, if you apply it smartly, can be just as powerful, or maybe even offer different insights than a super specialized complex algorithm built just for that one problem.
Absolutely. And like you said, it also gives us pause to think about standard procedures. That finding about log transformation potentially not being necessary for the raw Avanda data. It's a good reminder to sometimes question the usual way of doing things. Maybe simpler is okay sometimes, or different approaches can be equally valid.
So wrapping up our deep dive today, this research highlights tensor decomposition as this surprisingly powerful yet mathematically pretty straightforward method for analyzing complex genome-wide CRISPR screen data.
Its ability to integrate multiple sgRNAs and multiple experimental profiles simultaneously is a key feature.
Right, and it competes head-to-head with advanced methods like JSIC-S, plus it has that unique ability to work even without control samples by picking up on other biological signals like cell line efficacy, and it seems robust to sgRNA variation.
It really showcases the potential of taking established mathematical or data science tools and applying them creatively to tackle biological data challenges.
And that really leaves us with a final kind of provocative thought for you to mull over. If a method based on simple linear algebra can perform this well on complex biological screen data, offer these unique insights, and do it without needing controls or intricate modeling of things like sgRNA efficiency, well, it makes you wonder, doesn't it?
Yeah.
What other really complicated biological data sets or tough analytical problems out there might be effectively tackled or maybe even understood in a new light by applying fundamental mathematical techniques in novel, perhaps unexpectedly simple ways?